So good afternoon, everyone. My name is Carolina. I will be presenting today my latest work in my dissertation, where we focus on developing a system for battery-free recording of neural activity. So the title of today's presentation is Monitoring Evoked Somatosensory Activity with a Multi-Channel Passive Wireless Neurosensing System. My co-advisors are Dr. Volakis and Dr. Riera. So to start off with, we have a proposed system that has been the focus of many years of research with our different collaborators and the focus of my dissertation. The idea is to develop a fully implantable system that we can use to record brain electrical activity in a battery-free manner. The idea is to remove this battery because of the heat generation associated with it, the complexity involved in including a battery system in whatever implantable system you're using, especially in the brain, and the overall bulkiness that's associated with it. And overall, it's detrimental for our users and patients that would be needing the system. Our goal and our future applications include mainly epilepsy, but we have also considered potentially brain-machine interfacing and other areas. So to start off with, our proposed system, as I mentioned, is made up of an interrogator. This is meant to be placed directly on top of the skin or the scalp. On the other half, we have an implant. So both of these in between, we would technically, so we can't see this. Technically, in between the interrogator and the implant, we would have the skin in a completely closed implantation of the system. For simplicity and also understanding of the system, we just remove the skin there. So overall, we call this system the Wireless Neurosensing System, or WINS for short. Currently, it is about 40 by 40 millimeters in size, the implant and also the interrogator, with a goal in the future being miniaturization of the system. The biggest accomplishment as of right now, and the focus of today's presentation, is actually the multi-channel application of it, which involves two components, an optical component and a radio frequency component. So we start off with the optical portion. This involves having two LEDs on the interrogator to basically power and control a multiplexer in our implant. Multiplexer is simply a switch. We have another 3 LEDs in our interrogator, which are used for channel selection. So the idea is to switch the channels in the multiplexer of the implant at a very high frequency in order to be able to record simultaneously in up to 8 channels so far. Again, in the future, we'd expand the number of channels our system can function. So moving on to the radio frequency half of how the system functions, what we do is we supply a carrier signal at 2.4 gigahertz in order to trigger recording in the implant. Apart from supplying these optical signals, radio frequency signals, the implant itself has no power in it, has no function. It only works whenever we trigger it. That carrier signal that we supplied gets mixed in the implant with the output of our multiplexer in order to give us a modulated multiplex signal that can be wirelessly transmitted through the skin to the interrogator, where we process it and then finally end up with our 8 channels so far. In the future, maybe 32 channel recordings. Okay, so today's focus is we have 2 different components to it. First, we look at the benchtop testing of our system, since it's the first time we do multi-channel recordings. But we tried to set it up obviously in the lab, and then later on we'll see how the experiments worked out in vivo. As part of our benchtop testing, since we couldn't at the current size fully implant the system, what we did was we did a little bit of research and looked at a milk phantom to put between the implant and the interrogator to simulate a fully implanted scenario. Here we can again see all the different LEDs I mentioned, the 2 LEDs that would be used to power our multiplexer and the 3 used for channel selection. In the implant, we see the different photodiodes it communicates with. So basically, the gray LEDs communicate with the photovoltaic cell to power the multiplexer. The PD LEDs basically communicate with photodiodes, and those are just simply for channel selection. If we look a little bit deeper at the signal being supplied to the LEDs and then transmitted to the photodiodes and photovoltaic cell in our implant, we can see what we have here. The gray one is kept on constantly in order to keep the system on throughout the recording. And then the blue, the red, and the orange are switched at a high frequency in order to obtain whatever sampling rate you need for your recording. In the bigger portion of the figure, we can see what would be required to perform an up to 10 kilohertz per channel recording. The idea of this is to see what the limit of our system was. And it's set to 10 kilohertz, which is ideal for neuronal recordings, neuronal spike recordings, basically the activity of single neurons. In the inset of each one, we can see a larger pulse width in the signal being supplied to the LEDs, and this is used in order to perform 500 hertz recordings, which is also the focus of the in vivo portion of today's presentation. As part of our system, we also have an impedance matching network. This is used mainly to match the impedance of all of our wireless circuitry, our multiplexer, our matching network, and everything in our antenna system to the impedance of our electrodes. So a lot of electrodes have really high impedance. If your system has a much lower impedance than the electrodes you're using, what you end up is just reflecting your signal. Can't really record. So our impedance matching networks works mainly as an impedance buffer. It's just to match those two impedances. It brings the impedance of our system from our original publication, which was about 50 ohms, up to approximately 220 kilo ohms. We'll be working on improving this in the future. This impedance matching network does allow us to do at least a preliminary testing of the system. As we can see in the top part, we have everything in the implant up to the modulation. That signal gets transmitted, demodulated, and what we see on the other side is the modulated multiplex signal. These signals are then, each sample is decomposed in order to recover the 8 different signals that we see in the next slide. As part of our benchtop testing, we focused on 2 different components. In the time domain analysis of neuronal signals, we look at both frequency. We also look at amplitude. So first, what we did was use a regular signal generator. In order to emulate different frequency signals, these signals were selected specifically to cover the range of neuronal signals that we tend to want to record. So approximately from 1 hertz all the way to about 5 kilohertz. The lower frequency band looks at local field potential recordings, ECOG recordings, EEG recordings. The higher frequency band, neuronal signals. Then we have a side-by-side comparison where we see the signal we emulated with the signal generator and the signal we were able to recover with our system. Then we did the same thing, keeping the frequency constant and instead changing the amplitude. So we see a range of amplitudes from approximately 5 microvolts all the way to the millivolt range. And again, we can see how our system is able to pick up all 8 signals at the same time. We can decompose it and recover it. From there, we move on to the actual in vivo testing of the system. Our goal was to evoke different activity in the brain. We know the characteristics of these signals, so we know exactly what we were looking for and be able to conclude whether or not the system can actually record them. The selected sampling rate was about 5 hertz per channel for our system, mainly because since we were doing ECOG recordings, this is an ideal sampling rate. We didn't need to go as far as 10 kilohertz that we know the system can achieve. We used a 32-channel ECOG grid, which we can see right here in the bottom. Given that the grid has 32 channels and our system right now only works for 8, what we did was divide it into different groups in order to have an overall representation of what would happen if we were able to actually record 32 channels, at least in the future. On the top, we have an impedance characterization of the electrodes, where we can see that the electrodes have an impedance of about 40 kilo ohms at 1 kilohertz, well within the range of our designed impedance matching network. In the center, we can see distribution or the cortical distribution of the areas of the brain we're going to be evoking activity in, mainly the hind limb region, the forelimb region, and the barrel field, which is done by deflecting the whiskers of the rat. Then we can see the representation also in this last figure here. We use electrical pulses for the limbs and air puffs to deflect the whiskers. We look at a little bit at our results and actually the processing of them. We use typical filtering techniques in order to clean up the signal a little bit, remove 60 hertz. Bandpass filtered it up to 100 hertz in order to focus on the frequency component we need for ECOG recordings. Then we used ICA to remove any remaining artifacts in our signal. We epoch them, basically we divide them into segments and performed the averaging in order to extract our evoked potential. In short, what we would do with these recordings is we have, let's say, a 5-minute recording. We have specific triggers of when we stimulated the rat. We divide that entire signal into segments based on the stimulation and average. And that's what these red lines represent. The red line represents when I stimulated. The evoked potential is the activity that's elicited right after that. On this top portion, we can see the full 32-channel recording. Sorry, I forgot to mention. So all of these recordings were done with our system, WINS, and also a wired system in order to compare with. The wired system does have 32-channel capabilities. So on the top, we can see what the recording would look like if we were able to record all 32 channels at the same time. We have a heat map of how that signal is distributed on the cortex as time progresses. And then in the bottom, we only select the 8 channels that we can do at a time with our system and do an overlay just to compare them. This is specifically for what we saw as group 1 in the previous figure on the ECOG grid. and only for the hind limb stimulation. It's just an example. From here, we can see how the signal changes depending on which stimulation paradigm you're using. So in the first column, we again have hind limb. In the center, we have forelimb. And then in the last part, we have barofield or whisker stimulation. Each stimulation paradigm elicits a characteristic evoked potential. So that also allows us to make sure that what we're recording with our system is exactly what we should be recording. Just by visual inspection, we can see a very good comparison between both of them, but in order to really draw a conclusion from it, we did do some statistics. In order to basically extract specific parameters, we focused on 3 different things. Parameters typically used also for evoked potential analysis. We look at the peak, we look at the latency of that peak, and the overall width, or the full width at maximum of that peak. In order to pick these values, we used a Gaussian curve, with just the A in the function allows us to infer what the peak is. Then we have the amplitude, the peak amplitude, the latency, and the full width at maximum. The overall recordings that we see in today's presentation were as a result of 6 different rats or 6 different experiments. All of them were recorded with the wireless and the wired system, and the goal was to see if there's a statistically significant difference between what you would record with both systems. To start the analysis, we did a Shapiro-Wilk normality test just to see if our data was normally distributed. From there, we decided to work with a non-parametric Mann-Whitney test in order to test for that difference. And as we can see on this graph, we don't have a statistically significant difference in any parameter of any 3 stimulation paradigm, except for the whisker one. So in this case, our theory or explanation is that whisker, if we look back at the distribution on the cortex, a little bit farther away from where we place the grid for recording. So we can either attest this difference in full width at maximum in terms of the timing that can be elicited since our source is farther away from the electrodes than in other cases, or also in the switching capabilities of our system. So it is something we're looking at improving for the future, but overall we can conclude that we have comparable results with both of them. So concluding remarks, this is the first time we do test this system in vivo, so it's our initial validation of the system itself. We were able to record evoked neuronal activity and have comparable results with a commercially available wired system. We looked at 3 different stimulation paradigms: hind limb, forelimb, and whisker stimulation, and specifically at the key parameters we used for analyzing evoked activity, specifically the peak, the latency, and the full width at maximum. In the future, we are looking at expanding the system from 8 channels to 32 channels and overall miniaturizing it. Like I mentioned in the beginning of the presentation, it's about 40 millimeters by 40 millimeters. While it's a proof of concept, it would be better if we can bring the overall footprint of the system much lower. And that is the conclusion of my presentation. If you guys have any questions or any comments.